Good morning. Well, we're waiting for everybody to file in. I just thought I'd talk a little bit about your last molecule of the day. And so this is just something I came across for another course I'm doing. It's super interesting. It really shows the promise of so-called nanomaterials. And now that you all are starting to get to understand biomolecules a little bit better, now you'll be able to appreciate how these things are actually done. So as I'm sure all of you are aware, delivery of RNA into cells can be a very effective therapeutic thing. So the mRNA vaccines for COVID, mRNA is introduced into the cell with a either a viral vector or like some sort of a lipid nanoparticle, and then it directs the cells to make the spike protein, which the body mounts an immune response to, and away we go. But there's another way that RNA can be used therapeutically, and that is as something to silence the expression of certain genes. So you can get these things called siRNA, small interfering RNA. So they're small pieces of RNA that are complementary to whatever RNA target you're interested in silencing. And they form a duplex with that mRNA and cause its degradation. And so you can specifically tell the cell to not produce a particular protein. So for instance, ACE2 would be a good target for this in COVID. Anyway, so the way these work is actually anyway, so you can introduce siRNA through these lipid nanoparticles like you can for the Moderna or Pfizer vaccine, but when you want to silence a gene, you need much more efficient delivery than can be afforded by the lipid nanoparticles. And so these folks at the University of Connecticut invented these Janus base nano pieces. And so what they do is they basically package the siRNA into these, initially these nanotubes, and then they're fragmented into these nano pieces. And then these are taken up into the cell, and ultimately the siRNA goes on to silence a particular gene. Now look how these things are actually made. It's actually incredibly clever. So here's the basic unit here. So Janus, I don't know if anybody's interested in, I think it's from Greek mythology. It's kind of a two-faced god of some sort. Anyway, so this piece up here, it sort of looks like a nucleotide base, but it's actually kind of a fusion with two faces. And one side sort of looks like G, and one side sort of looks like C. And as we know, G and C come together to form 3 hydrogen bonds. And so these things can self-assemble in this way. So we get 3 hydrogen bonds forming between each kind of member here. And then if you turn this on its side, you get this sort of tube, because just like in a DNA double helix, you get this pi stacking of these bases. And so these nanotubes kind of spontaneously form. And the other part of this, the bases put the tube together, but the other part of it is a lysine that is attached to this. And of course, they drew it wrong here, the wrong ionization state. That's the story of my life. Every time I see something written as wrong. But anyway, so these can develop a positive charge. And as we know, nucleotides have a negative charge because all the phosphates. And so that is how the lysine residues allow the siRNA to be kind of packaged in these nanotubes. And then when they get into the cell, and this is also very clever, they're sort of engulfed by the cells, and they end up in these endosomes, which are membrane delimited compartments, and they got to get out of there somehow, right? So endosomes, normally, once they're formed, protons are pumped in to lower the pH. But because of all these lysine residues, the lysine residues act as like a proton sponge. So they soak up all these protons that are pumped in. And in response, the body pumps in all kinds of chloride ions, and then water has to come in to sort of equalize it all. And eventually the endosome pops and the nanotubes with the siRNA are released. So pretty cool technology. All right, so this is my last lecture to you guys. Dr. McLaughlin will be in next time. I just want you to know that I'll be maintaining my office hours up to the midterm at the end of the month. So don't be a stranger there. Today, I'm going to do a few iClicker questions. Haven't done them in a while, so you want to sort of get that ready. And so let's get started right now, actually. Okay. So we all remember from last time the mechanism of the serine protease. So the question is, which of the indicated reactions featured general acid catalysis? And I've sort of boxed the ones in question. So anyway, let's see how you do. Be a good self-test for your quiz that's coming up. All right, another 10 seconds. At least I'm getting some response, so that's kind of a good sign. 5, 4, 3, 2, 1. Stop. Okay. So acid catalysis is when something gives up a proton. So another way of asking the question, in which of these cases is a proton being given up, donated to something else? Okay, remember that, the two sort of phases of the reaction are parallel. So the right answer has got to be one from the left hand and one from the right hand. In fact, the correct answer is 2 and 4. So as we look, we can see that in both cases, histidine in this step starts off protonated, but then gives that proton away to the leaving group in both cases. Okay, so let's see how we all did. Most people got the right answer, or like the plurality was the right answer. So well done. Okay, that's promising. Okay, so let's move on to today's lecture. So we're going to finish off quickly these 3 remaining topics and enzymes, and we're going to start with Michaelis-Menten kinetics. And so there's certain terms that we need to understand, and it'll be Km and Vmax from this particular section. The textbook readings go into a lot more detail, especially on the derivation of the equation that we're going to look at. And we're not going to go through that derivation, but reading through that, I think, can enhance your understanding of the whole thing. But of course, that's optional. Okay, so why do we do enzyme kinetics? Well, enzyme kinetics basically is an experimental technique where you study how the rate of the reaction is affected by various parameters. And it's a very useful tool to help understand how a particular enzyme works. And the rate of the reaction will depend on many, many factors. And some of the ones that come into play here is how well the enzyme actually recognizes a substrate and how much of the substrate there is around. Okay, so this is kind of the general scheme. You've seen this thing at the top before. So again, the enzyme and the substrate interact reversibly through non-covalent interactions to form this enzyme substrate complex. Okay, so this is an equilibrium reaction on the left. However, a certain percentage of time, the enzyme actually acts on the substrate, converts it to product. And so if we follow the concentrations of all these various actors over the course of a reaction, when you first combine them, of course, you've got all free enzyme and all the substrate is initially there. However, as the reaction proceeds, gradually the substrate concentration goes down, it's consumed, the product concentration goes up because it's being formed. And at some point, you reach a point where the enzyme substrate concentration, so the concentration of this non-covalent complex is constant. Okay, it's not changing. It's just sort of turning over the enzyme, or sorry, turning over the substrate to product. And so this phase of the reaction we call the steady state reaction, the steady state phase of the reaction. And this is quite important ultimately for being able to model this whole process with Michaelis-Menten kinetics that we're going to talk about today. And in the course of deriving the Michaelis-Menten equation, which we're going to look at, a number of assumptions have to be made for the math to be, doable. And one of them is that the reaction is under steady state conditions. But that's the definition of the steady state. So the change in concentration of the ES complex is zero. Constant. It's a constant concentration. And if you plot the reaction rate, V naught here, against the concentration of substrate, you end up with a curve that looks like this. So this should be reminiscent of what we saw in hemoglobin. So again, this is a rectangular hyperbola. The equation is a little bit different, but that's the general form of the equation. And so as you increase the concentration of the substrate, initially the rate is quite low, and then it rapidly increases as you increase the concentration of substrate. But ultimately, there's so much substrate around that you reach a plateau in terms of how fast the reaction goes, and that is ultimately at an infinitely high concentration of substrate, you reach the so-called Vmax. Okay. And just like for hemoglobin, if you take sort of half of the Vmax, in hemoglobin it was half maximal binding, but if in this case we have half of the Vmax, and we extend that towards where the curve is and drop it down, and we get a constant. This kind of defines a constant. In this case, it's the Km. So very much reminiscent of hemoglobin. Or sorry, not hemoglobin, myoglobin oxygen binding. Okay, so the Michaelis-Menten equation is in the red box here, and it describes the relationship that we just saw. And it's named after Michaelis and Menten. Actually, Maud Menten was a Canadian biochemist and did some pioneering work over 100 years ago. Of course, she spent almost her entire career outside of Canada, because at that point, Canada was too backwards to hire a female to be a doctor or an academic. So we're a bit better now, but still a ways to go. If we look at this equation, the various components, V is the so-called initial velocity before any product is produced, and that's also known as the V naught. And so in deriving this equation, we kind of have to use this initial velocity idea. But in practice, it's very hard to actually measure that initial velocity, because as soon as you combine the enzyme and the substrate, then it starts reacting before you can even measure it. So I mean, if we look back at this thing, in theory, the initial velocity is down here, but in practice, what we end up measuring is whatever the rate is during the steady state, because this happens so fast, this initial increase to the steady state, that we can never capture it using standard techniques. So don't get confused by that. Okay, the Vmax is, of course, this maximal velocity. So that's when E is completely saturated with substrate. That's the concentration of substrate, of course, and Km is the Michaelis constant. Okay, so these are how these experiments are done. Basically, you set up a series of tubes with increasing amounts of substrate in them, and you add the same amount of enzyme to each tube and let them react for a little while. In this particular reaction, you get a blue color produced when the product is formed, and the more intense the blue color, of course, the faster the rate. And so you can plot the rate against the substrate concentration in this way. So the Michaelis constant, Km, is equal to the substrate concentration at 1/2 of Vmax. Okay, so that's very analogous to the P50 for myoglobin. All right, and in myoglobin, of course, the P50 reflects the affinity, or actually is the affinity for between the myoglobin and oxygen. Now, Michaelis-Menten kinetics, it's a little bit more complicated. There is the Km does not exactly equal the affinity or the dissociation constant, basically. Okay, there's another constant that's in there as well, but a greater Km translates into less affinity of the enzyme for the substrate. Okay, there's a little bit more going on there, but just like for a dissociation constant, there's this relationship between the value of the Km and the affinity for the substrate, and it's the same as the relationship between Kd and affinity. So a higher value means less affinity, a lower value means greater affinity. Okay, and one little bit kind of feature of Km that's somewhat important is that the Km is unaffected by the enzyme concentration. You could do this same experiment at a whole bunch of different enzyme concentrations, and in each case, you should get the same value for Km. Okay, so because we have this equation where the unknowns basically are the Vmax and Km, we can use the rate data that has been generated to figure out the value of Km and Vmax. Okay, and nowadays we'd use nonlinear regression using Excel. Even that useless program has nonlinear regression. You can plug in your rate and substrate data, ask it to fit this to this kind of an equation, and it'll spit out a Km and Vmax for you. But of course, in 1913, there was no such thing as Microsoft Excel. And so an easier way to derive these constants graphically was devised. And that's this double reciprocal plot, also known as the Lineweaver-Burke plot. And so this other equation here in red box, and we all know what that means, is actually the inverse of the Michaelis-Menten equation. But of course, it's of the form of y equals mx plus b. Okay, so y is 1 over v, x is 1 over s. So you're plotting 1 over v versus 1 over s. The slope m is Km over v max, and the intercept is 1 over v max. Okay. And so what does it look like? Here is a plot. So, okay, so of course, it's a straight line. And we can kind of draw the graph and get the values from the intercepts. So the x-intercept is 1 over v max. And the y-intercept is, the y-intercept is 1 over v max, and the x-intercept is minus 1 over Km. Okay. So it's much easier than Excel, at least for our folks in the last century. Okay, any questions at this point? Okay, so let's talk about enzyme inhibition next. Okay, so we're gonna, there's a number of different kinds of inhibition. We're gonna focus on only one, and that's competitive inhibition, because that's the, probably the most relevant and certainly the most intuitively easier to appreciate of all the modes of inhibition. Okay, so I mean, enzyme inhibitors are all over the place. There can be drugs. They can occur naturally to regulate various reactions within the body. Of course, herbicides and pesticides. Yes.
So in terms of like exam questions, are we going to provide with numbers? Are we expected to calculate KM? Or are you supposed to analyze it in enzyme KM? Oh, this one will be more likely.
All of the above. Know the equation, and then you're most of the way there. Okay, so in general, there's two different types of inhibitors. They can either be reversible or irreversible. And in a reversible inhibitor, of course, binds to the enzyme in a non-covalent fashion. And a good example of that is, for example, the statins, which are cholesterol-lowering drugs. I'm sure if many of you have parents that are on these things. And so these drugs bind non-covalently into the active site of an enzyme that regulates blood cholesterol, basically. Okay, and because the drug binds in the active site, the real substrate can't actually get in there. Okay, now because the binding is reversible, of course, the inhibitor can be removed, it can go away. But that's not the case with an irreversible inhibitor, which actually kind of permanently inactivates the enzyme because it forms a covalent bond with the target. And an example of that is sarin, which is a poison gas, basically, and it's been used in terrorist attacks and in warfare. And how it works is it targets this enzyme acetylcholinesterase. And by knocking out this enzyme, it basically paralyzes muscles. And so you basically suffocate because your breathing muscles are paralyzed. Okay, and the way this enzyme works, it's sort of reminiscent of a serine protease, actually. You've got a active site serine. It acts on acetylcholine, which is a small molecule rather than a protein, but the reaction is similar. So again, you've got a base that is around to take off this proton and turn the serine into a good nucleophile, but then it attacks sarin in this way, and then you get this intermediate here, but then the fluorine is lost, and you get this permanent adduct onto the active site serum. So this serine, so this enzyme is dead at this point. Okay, so competitive inhibitors are kind of the most interesting form of reversible inhibitors. So in a competitive inhibitor, the inhibitor actually binds to the active site. Okay, and so oftentimes you find that the competitive inhibitor looks like the substrate, looks like the product, or looks like the transition state. Okay, and of course, because the inhibitor is binding in the active site, it blocks access to the substrate. Okay, now as we shall see, the KM is affected by the presence of a competitive inhibitor, but the Vmax is unaffected. Okay, and so you could sort of see that you could actually diagnose the type of inhibitor, in this case competitive, by doing a Michaelis-Menten experiment and seeing if the KM or the Vmax is affected or both. Okay, and in the case of competitive inhibitors, the KM is affected, but not the Vmax. And the Vmax isn't affected because as you go to like an infinitely high concentration of substrate, because they're competing for the same site, of course, you can overcome the effect of the inhibitor at very high substrate concentrations. And so ultimately you would reach Vmax. And this is what the data actually look like. So again, we've got our direct plot on the left and our Lineweaver-Burke or double reciprocal plot on the right. And what we see with the inhibitor at any concentration of substrate, the rate is actually lower when you have the inhibitor there. Okay, makes perfect sense. Now, if you were to just look at these plots here on the left, ignore the right side for a second. If you're just going to look at those two plots and somebody said to you, hey, is the Km affected or is the Vmax affected? I don't know. It's kind of hard to tell just by looking at it. However, when you have competitive inhibition, there's a very specific appearance to the double reciprocal plot that is diagnostic. And as we see on the right, we have the two lines intersecting at 1 over Vmax, okay, the y-intercept, but the x-intercepts are different, which is -1 over the Km. Okay, so again, in competitive inhibition, the Vmax is unchanged, right? Because 1 over Vmax is the same, whether you have the inhibitor there or not. However, -1 over the Km is decreased. Therefore, Km is increased. Okay, when you've got the inhibitor there, that has the effect of seeming to increase the Km. And as we now know, when the Km increases, that's consistent with a reduced affinity of the enzyme for its substrate. And that should be intuitively, satisfying, right? Because you've got the substrate and the inhibitor competing for binding to the same site. So when you've got an inhibitor around, you're getting less binding of the substrate, and it makes it look like the enzyme isn't binding the substrate as well, because less is bound. Okay. Now, I'm not going to show this because I don't really have time, but different types of inhibition exist. In addition to competitive, you've got non-competitive, you've got uncompetitive, you've got mixed. And each of these different kinds of inhibition has a different diagnostic Lineweaver-Burke plot. Okay, so in some cases, the lines are all parallel. In some cases, they all intersect at -1 over Km, right? And so, this has a distinctive effect on these double reciprocal plots. Km and or Vmax are affected in different ways, and you can use these experiments to kind of diagnose which kind of inhibitor you've got. Okay. So, given your knowledge of the mechanism of competitive inhibitors, they come in and bind the active site. Given your knowledge of the Michaelis-Menten equation and the double reciprocal plot, knowing that in competitive inhibition, the Vmax is affected, but not the Km, you should be able to sort of put that all together, answer questions on what kind of inhibition is this, what happens with the Km, which of these two plots is the one that's actually in the presence of the inhibitor, questions like that. And there should be some of those on your practice quiz for this topic. Okay. So to summarize, enzyme reaction rates, of course, vary with the substrate concentration. More substrate, faster rate. And this can be described by the Michaelis-Menten equation, which has these two constants. And we can learn about the mechanisms of enzymes and enzyme inhibitors by doing these kinds of experiments. Okay, and it helps us develop enzyme inhibitors for various uses. And I'll give you kind of an example. Let's say you're working at a drug company, and you're interested in coming up with an inhibitor for a particular enzyme. If you based on your knowledge of biology, you know that if you were to inhibit this enzyme, you could reduce the growth rate of cancer cells or something, right? And so drug companies have these enormous libraries of compounds just sitting in their freezers and refrigerators. And they can draw on this library of compounds to identify which of them might be useful inhibitors of a particular enzyme. And so you've got your enzyme, you've got your substrate, and this whole library of inhibitors. Okay, and so what you then do is basically screen these inhibitors using Michaelis-Menten kinetics. Okay, so you vary the substrate concentration, you vary the inhibitor concentration, and when you put all the data together, you could identify which of those inhibitors is the most effective one. And you can identify the mechanism by which that inhibitor actually acts. Okay, so you get a whole bunch of different KMs if it's a competitive inhibitor. And obviously, you want to pick the one that increases the KM the most. All right, questions? All right, one last topic, and that's allostery. And this, when I'm talking about allostery, I'm actually going to harken back to the hemoglobin lectures, the hemoglobin lecture. And it'll also be a little preview of what you'll be seeing in the metabolism unit that's coming up with Dr. McLaughlin, because allostery is a very important feature of many metabolic pathways. Okay. So the reading in this case, they have a different example in the textbook of an allosteric enzyme that you can sort of read about. I like this example better, but some of the general principles are the same for the enzyme that I'm going to talk about and the enzyme they talk about in the textbook. So the word allostery comes from the Greek, okay, other place or other object, and the Wikipedia definition is the process by which proteins transmit the effect of binding at one site to another, often far away functional site, allowing for regulation of activity. And immediately you should be like, hey, that kind of sounds like hemoglobin, where you've got oxygen binding to one site, and then the affinity of the other three sites in hemoglobin is increased after oxygen binds to that first site. Okay, so hemoglobin is regulated. It's an allosterically regulated protein. It's not actually an enzyme, but allostery does happen in hemoglobin. And it can happen in enzymes too, as we'll see. And there's a lot of parallels between allostery in hemoglobin and allostery in enzymes. And so the hope is with these few slides that'll sort of reinforce the hemoglobin story, and also make it easier to understand this enzyme allostery. So hopefully the two stories will kind of mutually reinforce each other and really sort of drive home what I was trying to get at when I was talking about hemoglobin. Okay, so in allostery, there's a couple of different phenomena that can take place, often at the same time. Okay, so binding of substrate to one active site in an enzyme influences the binding at the other sites. So this would be cooperative binding of substrate. And that's what we see with hemoglobin binding oxygen. It binds it cooperatively. Binding of oxygen at one subunit of hemoglobin stimulates binding of oxygen at the other subunits. And the same is true of an enzyme that has cooperativity. Once substrate binds at one active site, then the substrate binds more effectively at the other active sites. Okay. Now, an important thing to remember is in the case of hemoglobin, the only reason why it is able to show this cooperativity is because it has more than one oxygen binding site. Myoglobin has only one oxygen binding site. It doesn't show cooperative binding. But hemoglobin has 4 sites. So you can have binding at one site and then a change in the quaternary structure and increased affinity at the other 3 sites. Okay. And it's the same with cooperative binding in enzymes. If you have an enzyme that is one polypeptide with one active site, you cannot get cooperative binding of substrate. So all enzymes that show substrate binding cooperativity all have more than one subunit and more than one active site. So the other thing that you can observe in allostery in enzymes is where you can have some sort of regulatory molecule. Okay, and this regulatory molecule is different from the substrate. Okay, and it can bind to the enzyme, change the conformation of the enzyme, and affect its activity. Okay. So the second phenomenon, again, fits in with this Wikipedia definition up here. Okay, so again, you've got binding of something to a particular site. There's a structural change in the protein that affects either its activity or its binding to substrate. Okay, so to really nail home, this relationship to hemoglobin, I've got another clicker question for you. Okay, and the question is, which of the following is not an allosteric regulator of oxygen binding to hemoglobin? Okay, so O2, BPG, protons, or maybe all of them are allosteric regulators. You decide. Ten seconds. Three, two, one. Okay. All righty. So, so let's take each of these in turn. Oxygen. Is oxygen an allosteric regulator of oxygen binding to hemoglobin? Anybody would like to take a stab at giving the answer and explaining why? Yes. Exactly right. So oxygen is an allosteric regulator of oxygen binding because binding of oxygen at one site influences the binding affinity at all the other sites. Okay, how about BPG? Yes. That's correct. Yes, it is an allosteric regulator because it binds in the central cavity of the T state, keeps the molecule in the T state, and decreases overall the affinity of binding to oxygen. All right, protons. Anyone? Yes or no, allosteric regulator? Yes. That's right. That's right. Very brave. Yes. It is an allosteric regulator. Remember, the Bohr effect is all about binding of protons to hemoglobin. When protons bind to hemoglobin, they stabilize the T state, kind of like BPG, and therefore reduce affinity for oxygen. And of course, that's important in the tissues where protons are high, they bind to the hemoglobin and promote oxygen release. And that's exactly what you want, because you want oxygen to be released in the tissues. Okay, and so most people got the correct answer here, which is D. All are allosteric regulators of oxygen binding. Okay, so keep all this in mind as we talk about this allosteric enzyme ATCase. So, I mean, this should be pretty obvious. This is a multi-subunit enzyme. It has multiple active sites. Okay, so binding of substrate to one of the active sites can increase the affinity of the other active sites for substrate. There's also some regulatory sites. Okay, and there's two allosteric effectors of this enzyme, ATP and CTP. Okay, and so I mean the active sites are sort of analogous to the heme groups in hemoglobin, right? So that's where you get the cooperative binding. And then the regulatory sites are actually analogous to where BPG binds or where the protons bind, because that affects the, the T and R state transition. Okay. Now, what reaction does this thing catalyze? So ATCase takes aspartate and carbamoyl phosphate, combines them into N-carbamoyl aspartate. Okay, you don't need to know any of that. Okay, all the scheme here, really don't need to know that. Why am I showing you this big pathway? So this is a classic example of a biosynthetic pathway that you see in metabolism. And many of these biosynthetic pathways, as you'll learn all about from Dr. McLaughlin, have many, many, many steps. Okay, so you start with relatively simple molecules like this. And then at the very end, you get your ultimate product, CTP in this case, or could be cholesterol, or could be one of the amino acids, whatever. All of these pathways tend to have multiple steps. And of course, the regulation of these pathways is really critical. You want to create the right amount of product in the right place at the right time. Okay, so these pathways are very tightly regulated. And that's going to be a major theme of Dr. McLaughlin's metabolism lectures. And what you'll find as you go through that is in a long pathway like this, usually the control is exerted at only one step. And it's usually a very early step. Okay, so in this long biosynthetic pathway, the rate of flux through the whole pathway is controlled mainly at one step, and that's the one where ATCase comes in and does this reaction. Okay, so ATCase has to be very tightly regulated because that will control the rate of synthesis of CTP in the cell. CTP, of course, being one of the nucleotides. All right, so this is CTP. It's not hemoglobin, or sorry, this is ATCase. It's not hemoglobin, it's ATCase, but ATCase also has T and R states. Okay, so the binding of a molecule of substrate, say aspartate, to the enzyme in the T state will cause the enzyme to transition to the R state, and that'll stimulate the binding of aspartate at the remaining active sites. Okay, so the T state has a relatively low affinity for aspartate, but the R state has a much higher affinity. Okay, so again, beautifully analogous to hemoglobin. And in fact, if you do an experiment where you vary the concentration of aspartate and measure the rate of the ATCase reaction, you get a plot that looks like this. So this enzyme is disobeying Michaelis-Menten kinetics because you're not getting a rectangular hyperbola. You're getting this S-shaped curve. Okay, and you can still derive a Km, but it's not Michaelis-Menten equation. And clearly what's happening here is at very low concentrations of aspartate, you've got the T state of ATCase, doesn't bind aspartate very well. But as more and more molecules of ATCase actually have one aspartate bind, then they switch to the R state and then they bind ATCase very well. And so you get this upward curvature and ultimately saturation near Vmax as the enzyme becomes saturated with aspartate. Okay, so again, very similar to the oxygen binding behavior of hemoglobin. In this case, it's forming a product. Okay. Now let's go back one slide here. And what's shown here is these green molecules bound to the T state. So these green molecules are actually bound to the regulatory sites. Okay, and this happens to be CTP. Okay, so CTP is bound in these regulatory sites, and CTP is acting very much like BPG or protons. It's promoting the T state. Okay, so CTP is promoting a form of the enzyme that doesn't work as well because it's stuck in the T state. And so if you have CTP around, what you see is that the curve here, the enzyme reaction rate curve, shifts to the right. Okay, the red line is with no CTP or ATP. As soon as you add CTP, it shifts it to the right. So this is just like the Bohr effect, or when you add BPG, you are, promoting the T state, which is a less effective form of the enzyme. You're shifting this curve to the right. Okay. Now, of course, things like this don't happen for no reason. There's a very good physiological biochemical reason why the enzyme might display this behavior. Would anybody like to guess at what that might be? Yes. Yep. Yep, that's exactly right. Thank you very much. CTP is, of course, the end product of this whole pathway that's supposed to be tightly regulated. As you accumulate CTP, then there has to be some way to turn the pathway off so that you make the right amount of CTP. And this is a phenomenon called feedback inhibition. Feedback inhibition. As you accumulate the end product, it feeds back to regulate the key step in its own synthesis, which is ATCase. And it does this by the allosteric effect. It promotes the T state by binding to those regulatory regions and slows down the enzyme. Now, ATP is a so-called positive allosteric regulator. It speeds up the enzyme. Okay. And I don't think in the case of hemoglobin, we ever identified a positive allosteric regulator like O2 and, or sorry, not O2, but protons and BPG are both negative. But in the case of ATCase, we have this positive regulator. So when you've got a lot of ATP around, that turns on the pathway. And there's a couple of different potential explanations for that. One is, of course, you need these nucleotides when you're, making RNA, when you're undergoing cell replication. Okay, and so under those conditions, you need to have a lot of ATP around for those molecules, for those processes to take place. And so the ATP here is kind of a signal. It's like, okay, we want to make lots of RNA, we want to make lots of DNA. There's plenty of energy around. We got to make sure that we have enough CTP being made to satisfy all these requirements. Oh my God, that's the end. All right, we made it all the way. Thank you. Thank you very much, everyone. Good luck on your quiz. Good luck on your midterm, the rest of this course. Stay on the forums. It's been a real pleasure. See you all around.